Welcome back to Dr. Liu's VCE Biology. Today's topic is DNA manipulation techniques and applications, as presented in the 2024 Victorian Certificate of Education, VCE, Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. A bacteriophage, virus, attaches to the membrane of pyogenes and injects its DNA into the cell. The CRISPR-Cas9 system defends against this virus by incorporating foreign DNA from bacteriophages as spaces between CRISPR repeats. This provides a memory of the virus, so if the same virus reinfects the bacterial cell, it can be recognized and targeted. Option A. The PAM sequence is located next to the target DNA, not the repeats in the CRISPR array. Option B. Option C is incorrect because DNA or genes are transcribed, while proteins are translated. Similarly, option D is incorrect. The Cas operant is transcribed to produce cRNAs, not translated. The CRISPR-Cas9 system in prokaryotes, bacteria, functions similarly to the human adaptive immune system, allowing them to remember and defend against specific viruses like bacteriophages upon reinfection. The canola plant is genetically modified, containing genes from cress plants, which means its oil is also considered genetically modified. Option B was a common incorrect response, as it reversed the genetic engineering process. It incorrectly stated that Agrobacterium recombinant plasmids were put into the cress plant, when in fact, cress plant genes were first inserted into the Agrobacterium recombinant plasmid before being transferred to the canola plant. Scientists in Western Australia have successfully used CRISPR-Cas9 gene editing to create new barley varieties, Hordeum vulgare, that are more efficient at absorbing and utilizing nitrogen. This innovation is expected to reduce the need for nitrogen fertilizers in agriculture and, consequently, lower greenhouse gas emissions, specifically nitrous oxide. The CRISPR-Cas9 technique modified a barley seed gene, such as HV1, through a single nucleotide substitution, which influences the chlorophyll and nitrogen content of the modified barley plants. The graphs show that the control group has a total chlorophyll content of 1.0 and a total nitrogen content of 0.2%. In contrast, the transformed group exhibits higher levels, with total chlorophyll at 1.5 and total nitrogen at 0.5%. The transformed barley significantly improves upon the control by boosting chlorophyll's function, resulting in enhanced light absorption, a more productive light-dependent stage of photosynthesis, and higher glucose yields. Additionally, its reduced nitrogen requirement can lead to benefits such as increased nutrient availability, better production of amino acids and nucleic acids, or improved protein synthesis. When using CRISPR-Cas9 technology, some barley seeds may not be transformed due to various factors. These include incomplete incorporation of CRISPR-Cas9 into all cells, failure of the gene to be edited, or unintended off-target editing, issues with Cas9 binding to the PAM sequence or cutting, problems with the single-guide RNA complementarity or mutations in the target gene, or the cell repairing the Cas9-induced cut without inserting the new nucleotide. Scientists extract DNA from barley plants and combine it with the BSTN1 endonuclease. This enzyme specifically recognizes and cuts DNA from non-transformed barley, but not from transformed barley. The resulting DNA mixture is then analyzed using gel electrophoresis to identify which barley plants have been successfully transformed. The transformed barley plant was identified as HV1c. This is because BSTN1 did not cut its DNA, resulting in a larger fragment visible on the gel, whereas it did cut the DNA of non-transformed barley plants. In gel electrophoresis, DNA fragments are separated by size. Smaller fragments move faster and further. Their movement is driven by their negative charge, which propels them towards the positive electrode. This section comprises 12 marks, 10% of the total assessment, which is 120 marks, distributed as follows. Short answer questions, 9 marks total. CRISPR-Cas9 in barley. Question 5. A barley application of CRISPR-Cas9 for enhanced nitrogen uptake and reduced greenhouse gas emissions in barley. Question 5A1. Impact of chlorophyll and nitrogen alterations in transformed barley. Question 5A2. Reasons for untransformed barley seeds via CRISPR-Cas9. Question 5C. Gel electrophoresis for identifying transformed barley. Question 5D. Principles of DNA fragment separation in gel electrophoresis. Multiple choice questions. 3 marks total. CRISPR-Cas9 system. Question 5. Representation and function of CRISPR-Cas9 in Streptococcus pyogenes. Question 6. Comparison of CRISPR-Cas9 function in S. pyogenes to the human adaptive immune system. Genetic modification in plants. Question 7. Genetically modified canola plants, herbicide resistance, and omega-3 fatty acids. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.